Hi, I'm Lincoln Stein. I'm head of adaptive oncology at the Ontario Institute for Cancer Research, and I'm happy to answer any questions you have about cancer research.
Hi, I'm Terry Hawrysh, and I do have a question. What are researchers looking for when they sequence DNA from tumor and normal cells of cancer patients?
Thanks for the great question, Terry. So when your doctors take a sample of tumor for genome sequencing, what they're doing is identifying changes in the tumor relative to your normal cells. So they'll take a sample of the tumor, they'll take a sample of normal cells, usually from a blood draw, send them both to the sequencing lab, and the sequencing lab will determine your normal sequence, the genome that you were born with, and the tumor's sequence, which usually has mutations which have caused it to become cancerous. And then what we'll be looking for are known changes in the tumor in genes which are treatable or otherwise druggable. So for example, patients with lung cancer who have changes in a gene called EGFR are sensitive to EGR inhibitors and will respond. Whereas patients who don't have this change shouldn't be treated with that therapy. It won't be effective. They'll be treated with something else. We also look for large patterns of mutations in the tumor genome. So for example, patients who have a large number of mutations are more sensitive to immunotherapy and are more likely to respond. The other things we look for are patterns of gene expression which are prognostic of the patient's tumor's aggressiveness, such that certain tumor subtypes which have a gene expression pattern that will indicate that they're more aggressive will receive more aggressive therapy. If you've consented to have your tumor's genome used for research, then we store it away in large databases of thousands or tens of thousands of genomes, and we use it to identify patterns that correlate the changes in the genome with clinical behavior to benefit patients in the future.